Hello everybody and welcome to our today's webinar about exosome analysis using a multiplex bead-based flow cytometry assay. We will start with a short introduction about exosomes and common methods to analyze exosomal proteins, with a focus on exosome detection by flow cytometry. Then I will introduce our multiplex bead-based assay, and in the second part I will show you results of exosomes analyzed from tumor cell lines and of different blood cells. Then we will have a short break with analysis of single exosomes by high-resolution microscopy, and in the end I will show you results of separation of different exosome subpopulations using immunomagnetic beads. So exosomes are defined by the release mechanism by the cells. They are secreted by outward budding of multivesicular bodies, and these are small vesicles about 30 to 100 nanometers large. And the release mechanism is different from microvesicles, which are generated by just outward budding of the membrane. Exosomes have been thought to be just waste of the cells, but since several years we know that that's not the case. So exosomes are important tool for intracellular communication and transport of material, as they can be taken up by recipient cells, either by ligand-receptor interaction or directly by membrane fusion or endocytosis. If we now have a closer look on the exosome consists of a lipid bilayer and is loaded with different nucleic acid, especially small RNAs, so microRNAs, tRNAs or ribosomal RNAs, but also mRNA and DNA. And in the membrane there are proteins. First to mention the so-called exosome markers, tetraspanins, CD9, CD63 and CD81. We will have a close look on them afterwards, but also receptors from the cell membrane like the EGFR receptor, different adhesion proteins, and also soluble proteins from the cytosol of the cells. Exosome composition depends on the cell type and the metabolic state of the cells, and can also be influenced by simulation of different cells. Therefore, we expect that there are a variety of different exosomes depending on cell types and the metabolic state. And of course, it's interesting to analyze these different exosomes. And common methods to analyze these exosomes are Western blot or mass spectrometry, where you can analyze the proteins. But the disadvantage of these two methods is that you see only the average of the exosomes, so you cannot get any information about individual or different exosomes, because it's just the mixture you can analyze. If you're interested in individual exosomes, people usually do electron microscopy, where you can see individual exosomes and can also detect proteins by immune staining. But here you can only analyze a few exosomes, and it's technically quite challenging. Another method to analyze single exosomes would be flow cytometry, and here we will have a closer look now. So the basic principle is that if a cell or an exosome passes the detection and the laser, it will generate a signal over time. And there are two different kinds of signals you can generate. The first signal is generated by light scattering of the cell by passing the laser, and there you can measure the scattering in forward direction and in side direction. The second signal, if you label the cells by a fluorescent dye, you can detect the fluorescence generated by the labeled cell. If you compare now the cells with exosomes, you immediately can see that the size of the exosome is about 100 times smaller than a cell. So the scattering of such small particle is hardly any. So you hardly can detect any scattering of the cells. But if we analyzed exosomes on the flow cytometry, you can see that you get a lot of signals in both channels, so forward and side scatter signals. But these are not single exosomes, but likely aggregates or swarms. You can detect the exosomes, but not single exosomes. The single exosomes would appear down here, and in this region is heavily overlaid by noise, and usually you use a trigger to avoid this noise, so you cannot see the single exosomes here. That these are really aggregates we proved by filtering the sample through a 100 micrometer filter. So we get rid of all larger particles and aggregates. And as you can see, after filtering in the analysis, we hardly see any signals anymore. So we lose the exosomes. So indeed, the detected particles are most likely aggregates or swarms. But still, it's possible to analyze the surface markers of exosomes by fluorescence detection. In this example, we have labeled the CD63 GFP positive exosomes with CD63 APC antibody. And as you can see, that all the exosomes can be detected by the antibody, and you see a nice staining. And this is also true if you're using two different antibodies. You can stain the exosomes with two different antibodies, so you can see that these two epitopes are present on the exosomes. However, as these are not single exosomes but aggregates or swarms, it's not possible to decide whether the two epitopes are present on a single exosomes or whether there are two different exosomes aggregating together and give the common signal. So you cannot address the question whether two markers coexist on the same vesicle. Therefore, you would need other methods, and we think that the multiplex bead assay could be one method to reveal such questions. I will now introduce you our new multiplex bead-based assay, and we developed this assay to simplify protein analysis on exosomes. The assay consists of 39 capture antibody beads, which are color-coded so that they can be distinguished by flow cytometry, and each bead is coupled to a different antibody. And with these antibodies, we can catch exosomes, either isolated exosomes or from cell culture supernatant. We incubate them together, and then we can use a different antibody or antibody cocktail to detect the bound exosomes on the beads in the flow cytometry. So how does this look like in the flow cytometer? We first do a gate on the forward side scatter to detect the single beads, and then we have a PE fits gating. Here you can see that we can easily discriminate the different bead populations by these two colors. And then for each of the detected populations or bead types, we detect the APC signal of the stained exosomes. How does the assay now look like in real? So this is a schematic drawing of 3 different kinds of beads. So you have to imagine that these are not just 3 but 39 beads, and we have different kinds of exosomes which can be bound to the beads depending whether the given epitope is present on the exosomes. And then we can stain the exosomes. We usually use a cocktail of CD9, CD63, CD81, so the common exosome markers, to stain all exosomes bound to the markers. And then we get a signal, and this signal can then be shown in a bar chart, so the signal intensity for each bead type. And in this case, you see that we get a signal for CD9 and for the CD63 bead, but no signal for the CD14 beads. So the exosomes comprise CD9 and CD63 on their surface, but no CD14. And as you can imagine, if you do a lot of these kind of analyses, it's a little bit tedious to analyze it on the flow cytometry. And as our PhD student Nina had to do a lot of such analyses, we developed an express mode for her, which allows an automated analysis. And this is shown in the next slide. So here you can see that the MACSquant flow cytometer automatically recognizes the 39 different bead populations, so it sets all the gates. And in the second page, it shows the gating and the results already summed in a table. So you get all the signal intensities calculated, and you can export the results for example, into Excel to generate the bar charts. We now come to the details of the assay. So we have selected 39 different antibodies. For example, the exosome marker CD9, CD63, and CD81, and also different markers known from different kinds of blood cells, but also some cancer markers. And we have 2 negative controls, meaning isotype antibodies, to estimate non-specific binding. Now, after introducing the platform, I would like to show you some results. And in the first experiment, we were interested in melanoma cells and their exosomes, and compare this to colon cancer cells. So we cultured the cells in the cell culture dish, harvested the cells, and then centrifuged it, took the supernatant and mixed it with our MACSplex beads, and with the staining antibodies, put it in the flow cytometer and analyze the results, and then we get the signal intensity for the beads. And we did the same with the colon cancer cells, so we could compare these two different kind of exosomes. And the results looks like this. On the x-axis, you see the different kind of bead types we have been using, and on the y-axis you see the median signal intensity we got for these markers. And on the left-hand side, you see these are the common exosome markers, so they have been present on all different kinds we analyzed. So we analyzed 3 different melanoma cell lines from primary melanoma and 1 colon cancer cell line for comparison. If you have a closer look, you can see that for MCSP, we only get signals for the 3 melanoma cell lines, but no signal for the colon cancer, so this is a melanoma marker. On the other hand, for CD326 EpCAM, which is not present on melanoma, and you could not detect it on the exomes as well, but you get a nice signal for the colon cancer. So we have now 2 markers which would allow to discriminate melanoma from colorectal exosomes according to these 2 markers. So I hope this has shown you the potential and how this analysis works. Now we are coming to a more complex example. We were interested in exosomes in plasma, and to know which kind of exosomes could be there, we isolated different cell types from blood, from healthy volunteers by MACS technology, and then cultivated different cell types in culture, and again took the supernatant, but in this case we isolated the exosomes by ultracentrifugation and then incubated the isolated exosomes together with the MACSplex beads and again stained them and analyzed them by flow cytometry, and we got an exome surface marker profile for each of the investigated cell types. And this is shown in the next slide. This is a little bit crowdy. Again, on the x-axis you see the different bead types. On the y-axis we are now using normalized signal intensities, because the signal intensity have been slightly different for the different cell types, because we are using different amounts of exosomes. And we were using the mean of the exosome markers CD9, CD63 and CD81 and set this mean to 100 to have comparable signal intensity to be able to show all the different markers. And as you would expect, we got signals for all the common exosome markers, so we had indeed exosomes. And if you are now looking on the different cell types, you see that, for example, CD2, CD8, CD80 and CD86 shows up for the T cell exosomes and NK cell exosomes. Then we have common markers which have been known for platelets, and indeed we found these platelet markers also in the platelet exosomes. In this case, we find these markers in nearly all cell preparations because they have been contaminated with little amounts of platelets. CD14 has been detected on monocyte exome, as you would expect. For B cells, we could detect CD20, and for modices, CD1C, CD11C, and CD13. And of course, we have also some markers which are detected not on a single cell type, but on several cell types like CD40 and CD86, which have been detected on B cells and modices. So we could detect markers which would allow to pinpoint exosomes found in the plasma to the original cell type. So if we compare now the results with plasma exosomes isolated from, again, healthy volunteers, now in a cluster analysis, in this case, each row, each column is one sample of different cell types and from plasma, and each row now comprises a bead type. And as you can see, the plasma signal is dominated by platelet exosomes, as you would expect, but you could also detect markers for T cells and NK cells, and for B cells or dendritic cells. So this data demonstrates that you can pinpoint exosomes from plasma to the different cell types, and that the plasma comprises mainly exosomes from platelets, but also from immune cells like T and NK cells, B cells, and dendritic cells. We had a closer look on the exosomes markers and were quite surprised that we find no CD9 on NK cell exosomes and hardly any CD81 on platelet exosomes. So these appear to be not general exosome markers, but they seem to be distributed differently on different exosomes. But we realized that this might be a nice chance to investigate different subpopulations of exosomes. So what would happen if we would mix these two exosomes? Can we then discriminate the two populations in our analysis? So in this case, we used NK cell exosomes, which are CD81 positive, and platelet exosomes, which are CD81 negative but CD9 positive, and mixed them together. If you would analyze these two populations by Western blot, as long as they are separated, you can easily see that the NK cells are indeed CD9 negative and the platelet are indeed CD81 negative. But of course, in the mixture, there's no indication that there are different kinds of exosomes in this mixture. So you just see that all markers are present, but you cannot see that these are two different kinds of exosomes. So how does this look like if we analyze the exosome mixture on the MACSplex platform? Now we are not using the staining cocktail, but single antibodies to be able to discriminate the two different subpopulations. So of course, as we have CD9 and CD81 positive exosomes, we bind exosomes to the respective beads, and if we now stain with the CD9 APC antibody, we only get a signal for the CD9. We only expect a signal for the CD9 beads and no signal for the CD81 bead. And if we stain with CD81, it's the other way around. We expect just a signal for the CD81 beads, but no signal on the CD9 beads. In contrast, for the CD63 beads, there are both kinds of exosomes should bind, so there we should get a signal for both kinds of stainings. So this is the expectation, and now I show you the real results. So on the x-axis, you see the different capture beads. On the y-axis, the detection antibodies we used, and on the z-axis, the APC signal intensities. If you now first look on the cocktail staining, you see that we get a signal for all three kinds of beads. So we get exosomes bound to all beads, as you would expect. But if we now look on the CD9 beads and the staining with CD81, we hardly see any signals. So there have been exosome captured on the anti-CD9 beads, but they are not stained with CD81 antibody. And the same is true for the CD81 antibody beads. Exosomes are captured there, but you don't get signal if you stain with CD9. So this means that we indeed could detect that in our sample, in a mixture, we have CD81 positive exosomes, which are CD9 negative, and CD9 positive exosomes, which are CD81 negative. So we could indeed discriminate the NK cell and the platelet exosomes in this kind of assay. If we now look on the CD63 beads, you see, as we would expect, that we get signal for all the different staining antibodies. So we would assume that we have double positive exosomes for CD63 and CD9 and CD63 and CD81. But the question arises, are these real double positive exosomes, or do we again analyze just aggregates? And this is quite a tricky question. And of course, you cannot prove this by the same assay. So we are looking for a different method to prove this, and came up by a method called high resolution fluorescence microscopy. So the principle of the assay is shown here. We were using a glass slide coated with CD63 antibody to catch the exosomes, and then we are using different labeled antibodies to stain the exosomes. And if the two markers would be present on the same exosome, they should be green and red at the same time, so should appear yellow. Of course, to prove that these are really single exosome, you need a sufficient resolution in the microscopy, and this is shown in the next slide. Here we compared confocal microscopy with the high resolution STAT microscopy, and as you can see in the first picture. In the confocal microscopy, you see just one large blot. And if you improve the resolution by that, then you see that these are 3 separated exosomes. So we can discriminate this. And this is also shown in the second example. Here we did a line scan where you could better see that we see 2 different signals in red for the higher resolution microscopy, but only one large spot in the confocal microscopy. So with the improved resolution, we could easily separate 2 different exosomes and show that that's not just a single event. And I have to add that we hardly find examples where these exosomes have been so close together. So most of the time they are separated and can easily be analyzed as single exosomes. So now we are coming back to the question, could we detect double positives, and are these indeed double positive exosomes? And for this we were using monocyte exosomes and stained them again with CD9 and CD81. And from the MACSplex assay we would expect that monocytes exosomes, at least some of them, are positive for both markers, and this could be proved by high resolution microscopy as shown here. We find some exosomes appearing in yellow, so having a staining for both markers. In total we find 17 to 27% of double positive spots on 3 analyzed image sections. And as a control we are using again the mixture of the NK cell and platelet exosomes. And here, as you would expect, we didn't find any double positive spots. So this proves that we can discriminate single from double positive exosomes, and that the double positive exosomes are real single exosomes comprising both of the markers. So this is nice that we now are able to find different subpopulations of exosomes. But of course, for downstream analysis, it would be nice to be able to separate these exosomes. And therefore, we tried our MACS technology, and we're using specific antibodies to label an exosome subpopulation, and then we can separate this subpopulation on a magnetic column from the other exosomes and enrich the exosomes of interest or the exosome subpopulation of interest. And as a proof of principle, we again used our NK and platelet exosomes, so you know this example now very well. We have seen this Western blot before, and now we used this sample and isolated them by CD63, and as both types of exosomes are positive for CD63, we get signals for CD81 and CD9. If we now, so we isolate basically the mixture, but if we now use CD9 beads, we only see a signal for CD9 but not on CD81. So we can separate the platelet exosomes from the remaining NK cell exosomes, and the other way around. If we are using CD81 beads, we only get CD81 positive exosomes and no CD9 positive exosomes, so we could separate the NK cell exosomes. So it's indeed possible to separate 2 subpopulations of a mixture of exosomes. Of course, this is just an artificial example, so we were looking for another example of a real sample, which would be more interesting and more challenging. And we came up with an example of primary B-cell exosomes. Again, we analyzed the exosomes on the MACSplex platform and used different B-cell markers for specific staining to stain for the B-cells. So again, on the x-axis, the different bead types, on the y-axis, the different staining antibodies, and on the z-axis, the signal intensities. And if you have a closer look, you can see that for the anti-CD42 beads, we only get a signal with this cocktail staining. And as we know, this is a platelet marker, we assumed that this signal is due to contaminating platelet exosomes, and we don't get any signal for a B-cell specific staining. So it's pretty clear that these are not B-cell exosomes, but contaminating platelet exosomes. But we realized that we get the same pattern on the CD9 beads. Again, only a signal for the staining with the cocktail, but no staining with the B-cell specific antibodies. So we assumed that CD9 is likely not present on the B-cell exosomes, but also due to the contaminating platelet exosomes. And to prove this, we decided to separate the two populations, and again we are using our microbeads, in this case CD61 microbeads, which are specific for the CD61 platelet marker. And this is how the original B-cell exosome preparation looks like. So we get signals for all exosome markers, we get signals for the platelet markers, and we got signals for the B-cell markers. If we now take the CD61 enriched fraction, so the eluate of the beads, you can see that all the platelet markers show up, and we could clearly decrease the B-cell markers. And if we now look for the flow-through, so the CD61 depleted fraction, we clearly could deplete all the platelet signals, and the B-cell signal is comparable to the original fraction. And now coming back to our original question, if we look on the exosome markers, you can see that the CD9 is present on the platelet fraction, but it's not, we didn't get any signal for the B-cell fraction. So indeed, the CD9 marker has been present on the contaminating platelet exosomes, but not on the B-cell exosomes. So the B-cell exosomes are the third exosome type, where not all of the three common exosome markers are present, but just two of them. So now we are already at the end of the presentation. I would like to thank you for listening. I wanted to show you today our new flow cytometry-based assay to analyze surface epitopes on exosomes. It allows to analyze 37 different epitopes in parallel, because you have two isotopes control. It comprises the common exosome markers, but also markers for immune cells or other blood cells and some cancer epitopes. You can detect the exosomes directly from cell culture supernatant, ascites or orine, or you can use isolated exosomes. I've shown you examples of single exosome analysis by high resolution microscopy to prove that we can discriminate between single and double positive exosomes. And I've shown you a few examples of immunomagnetic isolation, which allows the separation of exosome subpopulation for further downstream analysis. And during our analysis and the development, we realized that exosomes are by far more heterogeneous than assumed so far, and we expect that using such a method to analyze the different exosomes will reveal much more different subpopulations than known today. In the end, I would like to thank my co-workers who did most of the work in the lab. So Nina Kolia, who did her PhD work in my group, and Ute Heider, who developed the MACSplex beads, and Yvonne Winzek, who developed the exosome isolation kit. Eva and Ian helped us with the isolation beads, and for the exosome analysis of different bead cell types, we got different cell types from colleagues from other groups. And my special thanks to Astrid Schaus, Christian Jüngst and Nikolai Klatt from the CCAT in Cologne for the nice cooperation on the high resolution flow cytometry. And of course, thank you for listening.